Welcome to BioRenew Academy. Please like, share, comment, and subscribe my channel. I hope you all doing well on studies there. Today we are understand types of vaccines. Vaccines are classified based on how the antigen is prepared and delivered to the body, each type offering unique advantages for disease prevention. While understanding vaccine technology, vaccines represent one of humanity's greatest medical achievements, preventing millions of deaths annually. Each vaccine type employs different scientific approaches to train the immune system. From traditional methods using weakened pathogens to cutting edge genetic engineering, modern vaccinology offers diverse strategies to combat infectious diseases. 2. Live attenuated vaccines. The explanation of live attenuated vaccines. Here is a breakdown of the key points you can use to explain the topic. What they contain. These vaccines use a weakened attenuated form of the actual pathogen. This weakened form can replicate within the body without causing disease in healthy individuals. Key advantage. They offer strong, long-lasting immunity because they closely mimic a natural infection. Important note, due to the presence of a live, albeit weakened pathogen, these vaccines are not suitable for people who are immunocompromised. Common examples, Piscornistus generally or morbid reputation at diuterides PI, and common examples, origin from content on my mental try, and then common measles, mumps, rubella MMR vaccine, oral polio vaccine OPV, yellow fever vaccine, varicella chickenpox vaccine. 3. Inactivated killed vaccines. Inactivated killed vaccines are a type of vaccine that contains pathogens that have been killed or rendered inactive, so they cannot replicate or cause disease. How they work. They contain dead pathogens that cannot replicate but still trigger an immune response. The pathogen is killed using heat, chemicals, or radiation. Characteristics. Safe and stable for storage and transport. Can be used in immunocompromised patients. Generally fewer side effects. Limitations often require booster doses to maintain immunity since the immune response may be weaker compared to live vaccines. Examples. Inactivated polio vaccine IPVs, rabies vaccine, whole cell pertussis vaccine Panagat, 4. Subunit vaccines. Subunit vaccines, which are a targeted approach to immunization. Targeted approach. These vaccines contain specific parts of a pathogen, like proteins or antigens, rather than the whole organism. This focuses the immune response on key targets. Benefits. They typically result in fewer side effects and adverse reactions because only essential components are used, eliminating unnecessary pathogen material. Considerations. Adjuvant substances that enhance the immune response may be required to achieve adequate protection levels. Examples in practice. Hepatitis B vaccine uses surface antigen, human papillomavirus HPV vaccine, a cellular pertussis vaccine, 5. Toxoid vaccines. Toxoid vaccines, which are designed to protect against bacterial toxins rather than the bacteria themselves. Key details about toxoid vaccines. Mechanism. They contain inactivated toxins, toxoids produced by bacteria. The immune system is trained to neutralize these harmful toxins. Process. Bacterial toxins are treated with formaldehyde or heat to eliminate their toxicity while maintaining their ability to trigger an immune response. Examples. Tetanus toxoid vaccine, diphtheria toxoid vaccine pad. Important note, booster doses are required periodically because protection against toxins decreases over time. 6. Conjugate vaccines. These innovative vaccines link a weak antigen to a carrier protein, dramatically enhancing the immune system's ability to recognize and respond to the target pathogen. Weak antigen, bacterial polysaccharide that poorly stimulates immune system, chemical linkage, covalent bond connects antigen to carrier protein, enhanced response, strong immune response against both components. Why they matter, particularly effective in young children whose immune systems struggle to respond to polysaccharide antigens alone. This technology has significantly reduced childhood infections. Examples. Haemophilus influenzae type B hib vaccine, pneumococcal conjugate vaccine, meningococcal conjugate vaccine, 7 mRNA vaccines, a revolution. This infographic summarizes the key aspects of mRNA vaccine technology, highlighting its revolutionary approach to immunology. How they work: mRNA vaccines deliver genetic instructions to host cells, directing them to produce a specific antigen protein that triggers an immune response. Key advantages: this technology allows for extremely fast development, rapid adaptation to new virus strains, no live virus usage, and strong cellular bihumoral immunity. Challenges: they require ultra cold storage temperatures 20°C to 70°C and have a limited shelf life at room temperature. Real world impact: COVID-19 vaccines from Moderna demonstrated over 90% efficacy, with development completed in under a year. 8. Viral vector and DNA vaccines. These are two types of vaccine technologies: viral vector vaccines and DNA vaccines. Viral vector vaccines. This type of vaccine uses a modified harmless virus, like an adenovirus, to deliver genetic material that encodes an antigen. The vector virus cannot replicate but effectively delivers the instructions to the cells. Examples: Covishield AstraZeneca, Sputnik V, and the Johnson & Johnson COVID-19 vaccine. Benefits: generates a strong immune response and has single dose potential. DNA vaccines. DNA vaccines use plasmid DNA that encodes the antigen protein. This DNA is taken up by cells, which then produce the antigen, triggering an immune response. Examples: some experimental COVID-19 vaccines and various veterinary vaccines. Benefits: they are stable, easy to produce, and do not require cold chain storage. 9. Comparison at a glance. Comparison table that summarizes different types of vaccines, their key features, and examples. Live attenuated uses a weakened replicable pathogen, for example, MMR, OPV. Inactivated uses a kill pathogen that cannot replicate, for example, IPV rabies. Subunit uses specific antigen parts, for example, hepatitis B, HPV. Toxoid uses inactivated bacterium toxins, for example, tetanus, diphtheria. Conjugate links an antigen to a karyoprotein, for example, HIP, pneumococcal mRNA, provides genetic code for antigen production, for example, Pfizer, Moderna. ANA, viral vector, uses a modified virus to deliver genetic material, for example, Covishield, Sputnik V. DNA, uses plasmid DNA to encode antigens. Experimental. The choice of vaccine type depends on various factors, including the pathogen, target population, and storage requirements.